Right, all. Everyone should be nice and full now and hopefully not too sleepy. So we want to start our afternoon session, and I first want to welcome Pete Kelly, who is one of our sponsors from the Open Datasets Initiative, correct? And he is one of they work on government tech and policy affiliates coming together. So it's a novel public type data sets. And so we want to thank Pete for his support of the lab, as well as coming and telling us all about his exciting initiative today.
Thanks everyone for having me today. I'm sure everyone's a little sleepy after lunch, so I'll try to be pretty engaging here. Like I said, I'm Pete. I'm the director of the Open Datasets Initiative here on behalf of our nonprofit organization called Align to Innovate. My background's in synthetic biology, and most recently I spent about 10 years in industry building robotic work cells for high throughput strain engineering and running high throughput screening and assays. I'm really excited about the work that we're doing here because it's sort of trying to bridge a gap in capabilities, intentionality, and funding in what's currently available in industry versus what's currently available in the public sphere. So a little bit of background. Align is a nonprofit working at the intersection of biology, AI, and lab automation to improve life science with programmable spirit experiments. And the way we're going about this is by hosting a suite of programs with the overall goals of lowering the barrier to entry for using automation in the life sciences, both by creating validated open source methods and by starting to explore some natural language processing in the area. So, program the robots in English would be a really cool goal. We also, by doing so, want to democratize automation to help with the collection of large, high-fidelity, extensible life science data sets, which is, again, very valuable for machine learning and AI applications. And then, in order to enable both of those, we really need to think about how to create standards and benchmarks for biological engineering methods in this space. In 2022, Align hosted our first program called the Bioautomation Challenge. And this was the world's first grant program for promoting access to cloud labs. So we partnered with Emerald Cloud Lab, not the DAMP Lab, unfortunately, at that time, and awarded a cohort of 9 participants time on Emerald Cloud's resources to start writing foundational open source methods. This program is still in effect. They've been working on methods, and currently, we're in the process of starting to share those. They're hosted on GitHub. They're publicly accessible and available for other researchers to download. Please stay tuned for some publications, hopefully coming out of that in the next few months. And then building upon the success of the Bioautomation Challenge program, this year we've expanded the number and the diversity of programs that we're using to engage in the scientific community. So we are in the process of hosting a protein engineering tournament. Some folks in the room have helped organize the tournament. It was really a community-driven effort. And we took sort of our lead from the CASP program, which is the Critical Assessment of Protein Structure Prediction, GP in there too, which was a tournament hosted for the last decade or so, which set benchmarks for the community for enabling clear benchmarking for predictive methods for protein structure. This basically is what set the standard and really allowed for AlphaFold and RosettaFold and tools like that to, show their true power and really kind of set the pace for that. The protein engineering tournament is very similar to that, but rather than protein structure, we're focusing on protein function. This year's task is enzyme activity. And we, I think, had 85 or 86 people register for the tournament. Like I said, it's currently running. There's 2 parts, an in silico portion, which is wrapping up, and then an in vitro portion. We're actually going into the lab and creating and expressing some of these proteins and measuring their activity. That will wrap up near the end of the year, I think sometime in September or October. And then finally, the Open Datasets Initiative, which is the initiative that I'm the director of, and I'll be speaking about that for the rest of the talk today. The Open Datasets Initiative tagline is living datasets backed by automated open source methods, applicable to the folks in this room. We really came up with this to address a few challenges that we saw in the field. That for creating large datasets, there really hasn't been a venue for agenda setting. We want to, bring folks together to really list out what problems to address, what data sets are possible to collect, and which data sets are useful both for the biology community and the machine learning communities. In life science, many data sets are small, poorly curated, or privately held. All of these things hinder effective AI training and methods development, tools development for the public. And finally, the reproducibility crisis in life sciences. It's often very difficult to reproduce original collection methods well enough to expand. And if you are able to reproduce, results are qualitatively but not quantitatively the same a lot of the time. The inability to run these standardized experiments makes it extremely difficult to collect data and amalgamate it from multiple labs or multiple sites to build these data sets for machine learning applications. And industry, on the other hand, I think is pretty good at most of these most of the time, not 100% there. Industry has very focused funding, focused objectives, and they can create a plan to collect the necessary data, be intentional about the design of experiments, and then execute in a robust, predictable manner. And so we wanted to sort of take a similar approach to that. This is the kind of process that we've outlined for the Datasets initiative right now. It starts with soliciting ideas. We currently have an RFP open. It's available on our website. There's be a QR code later. Once we start to collect some ideas, really starting to generate community discussion with those ideas, setting workshops to target which data sets are the most useful, impactful, setting goals to try to benchmark, how would this data set be used? Is it really useful for industry? Is academia interested in it? Is there kind of broad interest? Is it very field specific? And also discussing the methods, the actual science for how we go about collecting these data sets, including standards, benchmarks, how do we be robust, how scalable are some of these automation methods that we're hoping to develop. And the goal through this whole idea surfacing and community discussion process is to generate as many ideas as possible. We really want this to be a very open field. We want to foster discussion with the machine learning communities, with the experimental biology communities, with the automation community to say, how do we build the right thing? How do we be intentional? And a great way to run a bad experiment is to collect data and then go to machine learning specialists and say, what can you do with this? Starting from the beginning with that intentionality to say, this is how we should go about collecting this data. This is the format. This is the type of metadata that we need is really valuable to being efficient through this process. Once we choose an idea that makes it through the selection process, we're going through methods development. The DAMP Lab is, like I said, is a partner, and we're hoping to develop a lot of these methods through the DAMP Lab to start making them accessible to the public. We want open sharing of scientific methods. So we really hope to host these methods on GitHub, allow them to be very accessible. Like, we don't want folks to have to comb through a methods section of a paper. We just want to clone a repo, download the method, and be able to execute in the same way every time. That also allows for interesting things like forking of methods or versioning in interesting ways that aren't currently allowable through traditional papers. And it also allows the data sets to be extensible. Like, if we collect data using a version of a method and a year later, 6 months later, another researcher says, I have more samples I'd like to process, that method still exists, is static, and can be used to add continually to that data set. Once the methods are developed, we understand our control structure, all of our best practices there. We go through the data collection phase. And we are subsidizing the cost of data collection here. That is the bulk of where our funding is going towards as part of Align to Innovate. Scientists around the world can submit samples for analysis. That data is available to them nearly immediately, basically as soon as it's coming off of the instruments, but will be embargoed to allow for filing of provisional patents or papers before being added to a community database, a publicly available database, about a year after the experiments are run. Again, the goal being creating public shared data sets, but we do have to create some incentive structure there to allow folks to actually contribute to these. And then again, that data will be released under a permissive license, which allows for its use in academia or commercial applications, depending on what you're trying to do. Like I mentioned, we're looking at a number of places for where this automated science will happen. Research institutions like the DAMP Lab, Carnegie Mellon potentially, EMSL, other members of the Agile BioFoundry. We've investigated some commercial labs. Like I said, we had a partnership with Emerald for the Bioautomation Challenge program. and then potentially industry as well. I think that's something we're still exploring. There's a lot of unused automation capabilities and expertise in industry, but the openness and sharing and publishing of methods is maybe a little bit trickier in that space. This is kind of a wordy slide, so apologies. Again, I just some notes on like how we're thinking about going about the experimental planning. Like we want to understand the downstream applications of the data that we're using. And so we're not going for perfection here. We're going for, good data is good enough, like I say here. If you're looking for a binary outcome on an assay, it doesn't matter if your liquid transfer CV is 2% or 20%. Understanding how the data will be used is crucial in designing an efficient, effective experiment. And on the other side of things, equivalency is very difficult to achieve, especially where biology intersects complex automation systems. So understanding your process, really characterizing it. And in my mind, equivalency means you can make the same decisions with the data, not that the data is the same. So to enable that, we're really being data-driven in this. We're trying to understand for each of these data sets, what are the standards and the controls for appropriate QC, performing design of experiment to develop robust processes and understanding where there is leeway or where there are failure points potentially. And then it goes without saying, metadata is incredibly important. Everything about which instrument it's being run on, the position within the deck, the lot numbers of reagents being used. All of this is very useful to back out the data on the other side of things and help make sense of it, to normalize variation, or even to understand where there's anomalies. And so through all of this, and almost out of time, we think we've addressed these three challenges that I mentioned earlier. With agenda setting, we are now creating a space for this discussion through our community engagement workshops for biologists, machine learning researchers, and automation experts to come together and talk about what is truly useful, achievable, and impactful. Using high throughput automation standards and controls, we're able to create large data sets that can be living and expanded over time. And using these open source and validated methods, and creating the right funding structures, we can start to chip away at that reproducibility crisis and really, again, build these large, robust data sets that are very powerful for advancing machine learning applications. So like I mentioned, we have a request for proposals open right now. It's a pretty lightweight process, a page or two. It shouldn't take you more than 2.5 hours to get something on paper. We're really going pretty broad here. If it is a data set that is useful to you as a biologist, useful to life sciences, we want to hear about it. So please do not hesitate to reach out. I stole my own line there, but yeah, what is the most impactful to your research? And how can we work as a community to enable data collection and curation at the scale necessary for emerging AI techniques? So if you're interested in learning more, feel free to visit our website, follow the QR code, or you can just email me at pete@alignbio.org. And I'd like to thank our sponsors, Schmidt Futures and Ken Griffin. Thank you.
Questions?
I was wondering, if you put a thought like how you're going to be getting data that's like similar data, but like data acquired by like different applications, like for example, like qualification data gathered from like a fluorescent phase assay, which is like this, how would you
Yeah, I think, again, it depends on the specifics of the experiments. I think the metadata of, what instruments, or even what particular instrument within a lab, tying that to the data being produced, even the metadata of what protocol was run or what, was the method that was used to generate this data point, is all going to be tied in. I think hopefully starts to address some of that.
For large multi-university projects, and then have you already been in discussions? Because a lot of times, if you have 8 labs from across the country who are trying to work on a common problem, it's how they amass the data together. Have you talked with groups like that?
We're in the process of doing that right now. This initiative only kicked off maybe 2.5 months ago. We finally got our webpage up and going. So a lot of what we're doing this year is trying to engage with the community, build our network of friendly folks and collaborators, and understand who's interested in what, and try to do some of that matchmaking.
Great,
thank you.
I just have it's more of like an open comment in the space. So Pete was just at a meeting in Maryland, along with Dave Vance from the DAMP Lab, related to metrics for this area. NIST, the National Institute of Standards and Technology, was involved. We have folks from MITRE through this BioMate effort. They have a project called Babel that's about bringing together data. there's the synthetic biology open language. What I think would be great, Pete, is in this process of you beating the bushes, I would love to see somebody, maybe your organization, someone else, a couple page white paper on just what's the state of the space. I mean, I was very engaged in this 2016, 17. That's now like ancient history. Like, what is it today in each of the? I think that would be valuable. And then to think about the last part as I finish my ramble would be, I would love some way that you would do some process, and it would be like stamped, like aligned, innovate, approved. Like, what would be the process? It'd be, it's heavy-handed, but I love that roadmap. If you want to be some compliance, that would be an interesting addition.
Yeah, I think you just summarized a lot of the topics of discussion at that NIST workshop. You know, there were basically, how do we, folks from MITRE were there, folks from SBAL were there, a lot of other folks as well from LabOp. And something that came out is, you know, how is it certified? Like, is there, you know, how do we agree on a standard or you know, a regulation of some sort to say this is an approved method, or this is, you know, guaranteed to do X, Y, or Z. And I do like the idea of the white paper or some state of the automation address or something like that. I'm hoping that shakes out of the EBRC and at NIST workshop, but I know that's maybe still 9 months out or something like that.
And I'd be happy with Betamax versus VHS right now. Like, I would be happy if they were competing standards.
Yeah.
And then we kind of saw what happened. I'm not saying we have to pick one before we start.
Yeah.
But at least that would, like, VHS was defined, Betamax was defined.
Yeah, we'll be the Blu-ray of synthetic biology, basically. Yeah. Tracy.
I'm just going so this might be a great topic for a workshop. They say, like, what is the white paper?
Yeah, so
Tossing it up.
yeah, let's do that. So I'll keep talking.
Tracy was also at the NIST workshop.
I had mentioned what's going to happen with this IWBMA workshop next year will be in September. That will line up with this regular occurring Biodesign Automation Week. But there will be one this year. This workshop will be there. It's happening. But again, Tracy is going to be the general chair of the International Workshop on Biodesign Automation. Sounds like we just posted.
We just put out the policy proposals for IWBMA and all the workshops for the week itself.
It sounds like I have some homework then. Great. Thank you, everyone.
Yeah, great, Pete.